Okay, hello everyone. We'll start with the bioinformatics part of this webinar. Okay, so just to recap what we have done in our first session. We have introduced to you about the Amplicon sequencing that is a part of NGS where we targeted on certain genomic region. For example, we have 16S V3V4 or 18S and ITS regions. Then once we have completed the sequencing, now we can start with the bioinformatics analysis. Just a quick introduction for bioinformatics analysis. So bioinformatics analysis is actually the analysis of biological data using different computational tools and often requires programming languages as well. And at the end of bioinformatics analysis, we can obtain statistics, graphical results for better presentation of our analysis. So now let's start with the bioinformatics workflow analysis for Amplicon sequencing. Now, there are four main processes in Amplicon analysis. So the first one is the quality assessment, where we want to check the quality of the sequence and data that we have. Next is the distinct reads selection, where in this process, we want to get the specific reads using different methods. And we will explain it in further next. The third process is the taxonomy profiling, where we want to assign taxonomy to each distinct reads that we have. And the last one is the statistical analysis, where we want to mainly compare the abundances that we have obtained from our samples. So now let's get into the first process, which is the quality assessment. So for this process, this part is actually depending on your sequencing run. Usually we will do these different 4 steps. So the first one is the adapter and or primer trimming, meaning that we want to trim out any adapter or any primer that we use in our sequencing that which is specifically for regions that you have chosen. And then next, we want to trim out any low quality reads from our samples. Usually we will do trimming for quality below than 30. And the next one is the region length filtering, meaning that we want to focus only on the region, the length region that we have focused. So now we know that, for example, 16S V3V4, the length is around 460 base pair. So we will filter out any length that is lower or higher than that, so that we can focus only on this region. The next one is the Chimera screening, meaning that Chimera is actually the artifact sequence, something like a fake sequence that is formed when 2 biological sequences merge. This can be formed during PCR process, and it is actually common to have a chimera in your samples because this happens when closely related sequences are amplified. So we also want to remove this sequence out as well before we proceed with the next steps. Now, once we have done all of this, we can go to the next process, the distinct reads selection. Distinct reads selection is actually we want to get any distinct or unique sequence from our sample. We want to find we want to find the unique sequence that are different from other reads. So there are, in general, there are 2 types of methods here. So the first one is the operational taxonomic unit, OTU, or we'll call it as Otu Clustering Method. So in this method, it clusters reads that are based on similarity threshold, usually at 97%. And there are a few types of Otu Clustering Method, which are de novo, closed references, and open references. So on this slide, I just want to show you how the concept of the Otu clustering. So for example, here we find this one sequence, and then we will take this as one OTU. So, and then next we can see another sequence that are almost similar but not exactly the same, because we can see that there is a difference at this position here. And then when we calculate the similarity of this sequence to this sequence, we can see that the similarity is around 99%. So we will take this as the same OTU. So this sequence we will also consider as OTU1 as well. Okay, so next we find another almost similar sequence, and we can see that there are some differences here. So when we calculate the similarity, and we can see that the similarity is around 95%. So now we know that this sequence is actually not the same one as OTU1. So we will consider this as OTU2. So these two OTU is different, okay? And the same goes with the next sequence. Once we calculated the similarity, it is around 74%. So it is definitely lower than these two. So we will also consider this as another OTU. So basically, this is how Otu clustering method works. The next method is Amplicon's sequence variant ASV method. So in this method, it's actually expect to have the biological sequences that can be observed more than the error sequences. And in this method, they only have de novo method, and they also contains a process called denoise, which is actually an error correction process. So they will do some correction here before they proceed with the calculation. So the same as previous example. So right now we have this one sequence, and then because ASV they can detect up to one nucleotide differences. So for example, here we know that this position is different than this one, right? So because of this, and also once they have checked on the biological sequences, we can say that this will be another ASV unit. So this one is the ASV1, and this one will be considered as another ASV2. The same goes for this one. For the next sequence, we can see that there are difference in this position. So this will also considered as another ASV unit, which is ASV3. And as well as this one, we see that these positions here are different. So this can be considered as another ASV unit. So that is how basically OTU and ASV are different. Okay, once we have done all of that, we can now assign the taxonomy to the reads that we have found before. So in this step, we will have different database. We can assign these reads to a reference database that we want that we choose. So usually we will go for Seluruh database, but we also have the other databases as well. For example, Green Genes, RDP classifier database, Unite database. But for Unite database, it is specifically for ITS region. And we can also curate our own reference database from NCBI database. But most importantly, we need to remember that no matter which database that you choose, you need to curate that. You need to make it into a specific region of interest that you want to that you have for your samples. So at the end of this process, you can have something like this table. So here we have the ASV ID. So this one will be the ID of the ASV that we can find in our samples. And then this is the abundances, the number of times that you can find this sequence. So for example, here, this the first ASV here, you can actually find it 316 times in these samples, and so on for these samples. And then we can see that, okay, so now this ASV, this taxonomy is this this line here. So it depends on the database that you choose, okay? So once we have this that table, now we can proceed with the statistical analysis. So for the statistical analysis, basically we want to compare the abundances between samples or between groups. So most of the time we will use the table, this table here for the statistical analysis. Okay, so for the diversity, for the statistical analysis, we will have different analysis. So for the main one is the diversity analysis. So diversity analysis is actually to compare the abundance between samples or between groups, and diversity analysis can be categorized into alpha diversity and beta diversity. So alpha diversity meaning that you want to check the diversity in a local community. And for beta diversity, it is between communities. So for example, let's say that this one box here is one sample, okay? So now in this site A, we have these few samples. So for alpha diversity, we want to check only in this site A only, the diversity in site A only. So for beta diversity, we want to check between sites, meaning between site A and site B. So that is what it meant by alpha and beta diversity. So for diversity analysis, there are a few indices. So these indices is actually quantitative measures to show differences in communities depending on the formula that they use. So for alpha, usually you can see these words here in any Amplicon reference paper, which are richness and evenness. Richness is to check the number of organisms, how many different organisms that you can find in your sample. So for example, we have Chow-1 calculation or Shannon. And for evenness, we want to see how these organisms are distributed along your samples or along your samples. So for example, we have Simpsons or Robbins. So this part is depending on your research objective. What do you want to check? Is it you want to see how many organisms that you can find, or you want to see how this organism is distributed in your samples? Next one is the beta diversity indices. So in beta diversity, it is either to check the abundance of this organism are similar or different in terms of counts or of organisms, or whether the organism is present or not. So for this beta diversity, we have here Bray, Curtis, checkered, and Unifrac. So this is also you can check how they calculated this to know whether it is suitable for your project or not. Another interesting diversity analysis that you can do is the Venn diagram, meaning that you want to compare the abundance of unique organism in your sample or your organism. Okay. So now let's see how it looks like. The example of the output for this diversity analysis. So here you can have something like this one, like a dot plot where you can have the value of each of your samples here, or you can also have a heat map here from the weighted Unifrac. And this one is the Venn diagram analysis that I have mentioned. So like in this one, because as I mentioned before, it is for unique organism, right? So for example, here you know that there are 1,077 ASV or OTU that can be found only in this group. and 271 organisms that can be found in group 2 and group 1. So that is how you check it. Okay. So next is the taxonomic statistic, meaning that from the table that we obtained before, we can also generate graph so that we can see what is the composition of each samples. And usually we will also want to focus on the top 10 organisms that can be found in each taxa level. So you can have something similar like this bar plot here. And the next one is any advanced analysis. This is very suitable if you have a specific objective. For example, pie crust analysis. Pie crust analysis is actually to predict functional pathways that can be found in your sample based on the sequence that we obtained before. And then next one is Corona. Corona is actually a visualization of your taxonomy profiling. So in here, you can also search for any specific taxa, and then they will show you in this pie chart with the percentage as well. Next, you can also do correlation analysis, where this is to correlate the characteristics of your samples, or if you have any metadata like the pH of your samples, the condition of your samples, you can use that metadata to map that against your abundance found in your samples. Again, this is optimum requirement for the optimum setup. You can have more than up to 32 gig, 8 cores of CPU and storage 50 or more gigabytes. But this is also depending on the number of samples that you have. Whether if you have 1 or 2 samples, then you can go for the minimum requirement. Or it is also important to know the complexity of the tools that you want to choose for your analysis as well. Okay, then I will show you example of bioinformatics analysis report that you may get. So for example, this is our first phase bioinformatics analysis Amplicon report. So for example, you can have an overview and the workflow of each of the processes that you take. And then next, you can have a sequence QC report where we put the QC report of all of the samples. And then you can have the bar plot of the taxonomy assignment that based on the ASV table. And then you will have something like this one, the diversity analysis graph here, and then you can have the beta diversity as well here, and then the Venn diagram. And you can also do phylogenetic tree. For example, here we have the top 30 genus. So this can also be considered as the advanced analysis. So here we have the phylogenetic tree here. And then for the first base NGS service here, we also provide you different services, as you can see in this slide. You can opt for different raw reads as well. And we also, for our own BI analysis service, we have this in-house analysis service that use DA2 pipeline. And this pipeline is suitable for these regions here. So if you have, if you want to know more about our bioinformatics analysis, you can always let us know and email us. Okay. We also provide other Amplicon services as well. For example, we do provide a whole genome sequencing, whether it is for if you have your reference genome or if you want to proceed with de novo analysis. We also have WES for exome sequencing, RNA sequencing, and such so on. So just let us know. And other NGS related services as well. For example, we do DNA or RNA extraction as well as purification. So this marks the end of this slide, the webinar. And just to conclude, we have introduced to you the application of next generation sequencing in research fields. We have also showed to you how you can use Amplicon sequencing to detect and profile the abundances of bacteria in your samples, and as well as the bioinformatics workflow where you can use it to analyze the Amplicon data that you have. And there are also many different ways to utilize the data that you have, either by statistical analysis or via visualization in bar charts and graph. But however, I just want to make it emphasize on this again, that it is important for you to know that you need to focus on your research objective before performing any bioinformatics analysis, especially for Amplicon sequencing. So it doesn't necessarily that you need to do all of this analysis. Sometimes, because as I said before, it is depending on your research objective. So if you have any question or anything that you want to discuss with us, just let us know. So for now, I can pass back to our host, Irene.
All right. So thank you so much, Afrina. Thank you so much, Afrina and Kitley, for the insightful webinar. So we have been receiving a lot of questions from you all, and we have a team that is answering them. Now, for majority of your questions, some of you all were asking on projects. All right. So you have said some of your interests, and you have also asked based on projects. So some of these projects can be customized as well. So for those of y'all that asked, or basically we have replied that you can get back to us through NGS. We have written an email address there. So NGS at Apical Scientific. So we hope that you would write back to us. On the other hand, we have also few questions. Now, for the rest of y'all who have asked questions, but we are still getting the team to answer. So if you do not get your answers here, so we will be sending you an email, right? So you can continuously continue to ask questions. All the questions asked by our participants, or basically our attendees here, you can always check it out at the question tab. So you have a list of questions that is being asked, and we have answered accordingly. And for some of them, we'll be addressing them through private emails. All right. So thank you so much for joining us. All right. So if you have any other questions,